Welcome back, AP Bio, as we dive into Chapter 17 and talk about viruses. So we need to just start off with, what is a virus? I know there's a lot of confusion about virus and bacteria and pathogens, and we can even debate whether or not they're even technically alive. So a virus is an obligate intracellular parasite. So let's break that down. Obligate, it's required. Intracellular, which means within the cell. And then we know that a parasite is something that lives off something else to the host's detriment for its benefit. So it's required to parasitically live within cells. And when you break down what is a virus, it's basically just some genetic information, so either DNA or RNA, surrounded in a protein coat, which is called a capsid. And that's it. Now, some viruses are going to get a little bit more fancy. They may have a membrane around it, and they may have little glycoproteins that allow them to trick immune systems, but at its core, it's just DNA or RNA, genetic information, and a protein coat. A famous virologist, Sir Peter Medawar, once said that a virus is a piece of bad news wrapped in a protein, and I absolutely love that definition. So viruses are sort of the living dead of biology. So they cannot carry out their life cycle independent of a host cell. They're obligate intracellular parasites. And they're not made of cells. They're just protein and nucleic acid. And because they can't replicate on their own, and they don't metabolize anything, they don't have little mitochondria, they're not making energy, they're not considered alive. But they are able to reproduce. They have a huge impact on biodiversity. They're made of biomolecules, and so they kind of live in this nebulous gray area. And viruses may be sort of a good view as to the origins of life. So there's a lot of famous viruses, and viruses have a huge impact on our world. You can go back to the 1918 flu pandemic, the herpes virus, measles and polio, and smallpox, which we've been working to eradicate, some of which have been making a comeback, like measles in recent years. And then I would be remiss to not even mention our current state of the world, which has been completely turned upside down by the novel coronavirus. So viruses have a huge impact on our life. So how did we first discover viruses? Well, that goes to Dmitry Ivanovsky, and he discovered viruses while he was researching the transmission of a plant disease called tobacco mosaic virus. And he discovered that viruses had to be smaller than cells. So at the time, they thought it was like a bacteria, some sort of cell that was causing the disease, some sort of pathogen. But what he did was he took plants that were infected with this tobacco mosaic disease, and he took sap out of them, so the liquid within those leaves, and he passed it through a filter that was known to trap bacteria. So bacteria would have gotten stuck in this filter. Then he took what went through the filter and rubbed it on healthy plants, and those plants became infected. And so this proved that there was something smaller than a cell that was capable of reproducing and causing harm. And so this was our first clue as to a virus. So there's viruses that affect every living cell in the world. And one of the most iconic looking viruses are phages, which are bacteriophages because they attack only bacteria. Now, these are the #1 mass murderers in the world. They kill more life in terms of just raw mass than any other organism on the planet. They destroy bacteria. They live to destroy, or maybe not live is the right word. And so they're known as phages, which comes from the Greek word for consume. And this is because when phages were introduced to fields or lawns of bacteria in petri dishes, they would just wipe them out. And there are estimated 10 to the 31 power bacteriophages on Earth. This is more than the number of stars in the known universe. If you lined up these tiny little bacteriophages, it would stretch up to 100 million light years long. So there's just an absurd amount of phages on our planet right now, wreaking havoc on the micro universe. And in fact, because they're so good at their job and they only attack, viruses are very specific to what type of cell they attack. So bacteriophages can be all over our bodies and they won't do anything to our cells. And so bacteriophages are being used in experimental treatments of bacterial diseases that might be resistant to antibiotics because phages are so specific to bacteria. And this type of treatment has actually been used for around 80 years around the world as a treatment for different bacterial infections. Just not commonly used in the United States yet, but maybe. So how do these viruses reproduce? What's their process? So we're going to explore two types of processes, the lytic cycle and the lysogenic cycle. So in the lytic cycle, and we notice that prefix lytic is going to sound like lysis. The bacteriophage is floating through the world and it happens to land on a bacteria and it's going to attach itself. Then that bacteriophage is going to inject or insert its genome, whether it's DNA or RNA, into the host. That then is going to provide the information to start assembly of more viral proteins. And so basically this bacteria gets hijacked and all its cellular machinery, all its ribosomes, everything, no longer are doing what the bacteria wants, but doing what the virus wants. And it's building new bacteriophages. And eventually this cell is going to get so full of bacteriophages that it's going to lyse or it's going to burst, and all those new bacteriophages are going to be released into the world and they're going to infect more bacteria, and this cycle continues. And so there's an alternative formula for this called the lysogenic cycle. And the lysogenic cycle is where you have the bacteriophage attaches again, it inserts its DNA, but instead of immediately taking over the cell, that DNA gets integrated into the bacterial DNA and it just waits. And so it enters this lysogenic cycle. And that bacteria reproduces, and when it copies its genome, now both daughter cells are going to have, at the end of that binary fission process, both daughter cells will have that viral DNA. And as it continues to replicate and reproduce, now that viral DNA is spreading and spreading and spreading and spreading. And eventually, when circumstances are right, it shifts from this lysogenic cycle over to the lytic cycle. And that viral DNA activates, it takes over all the machinery inside the bacteria, starts producing a bunch of new viruses, and then it's going to lyse or release all those bacteriophages to infect new cells. Now, the lysogenic phase is really cool because sometimes it doesn't enter the lytic, and these viral genomes just get stuck into DNA and they linger there. In fact, the human genome, it's estimated around almost 10% of our DNA in all of your cells is actually the leftovers from old virus infections, old viral infections that got inserted into the DNA, never took over, and are just lingering there. So if you are your DNA, you're about a 10th virus. So here's a nice little GIF that's going to show us the difference between the lytic cycle and the lysogenic cycle. So in lytic and lysogenic, they both get infected. In lytic, it starts reproducing immediately and they burst, whereas in lysogenic, it stays in the DNA and as every time the cell divides, it now gets spread to those new cells. Eventually, it'll go back to the lytic, where infection, and immediately make new viruses until it bursts, killing the bacteria, releasing millions of new viruses. So those are bacteriophages or bacterial viruses. What about viruses for eukaryotic cells? These are actually way more diverse because eukaryotic cells have a much more robust defense system. So it could be that it's part of a multicellular organism where there's specific cells that target pathogens like viruses, or just the organelles. There's all sorts of proteins and organelles whose whole job is to protect the cell from things like viruses. So as a result of this, the viruses that infect eukaryotes have to be a whole lot more diverse. You'll see both DNA and RNA genomes. You'll see both double-stranded and single-stranded RNA. You'll see both double-stranded and single-stranded DNA. And you'll find that most have a lipid envelope that surrounds the protein. And that's because instead of causing those cells to burst, you're going to see that those viruses are going to bud out, and they're going to get coated in the host cell's membrane. That's going to give it that lipid envelope. And now that it has a host cell envelope, it can hide in that host organism because it looks like one of the host cells. So very sneaky. So one of the classic examples is influenza, or what causes the flu virus. And this is an RNA virus, which means RNA is its genome. It's especially nasty because it's easily spread through the air. And influenza is actually responsible for more deaths in human history than any other known virus. So this is like the #1 viral killer of people, to our knowledge. And so how does this work? Well, that viral RNA, that virus, is going to get absorbed into the cell. Remember, it has a coating that looks like it belongs to the cell, looks like it's a friend. And that viral RNA is going to be released and enter into the nucleus. Then what it's going to do is prevent the host cell's mRNA from being read, and only viral RNA is going to go to those ribosomes. So it's going to start manufacturing viral proteins. So the viral RNA and the viral proteins are going to get sent to the Golgi apparatus. Remember, that's our post office. And it's going to get packaged up and sent to the cell membrane, where all those packaged RNA and proteins get pushed out, exocytosis, and it's going to get wrapped up in that little envelope of host cell membrane, and it's going to go on its way, and now it looks just like a host cell, and so it'll get accepted by new cells. It's going to look like a friend instead of a foe. Because it's an RNA virus, there's no proofreading mechanism, and so that means that the viral RNA is going to mutate really, really rapidly. This is known as antigenic shift. And so this is what makes the flu virus so difficult, why you don't get immunity from one year to the next, why vaccines are so hard, because the virus is constantly mutating and constantly changing. And so the virus you see one year is different than the next. It could be the virus you see 1 week is going to be different from the next. And so this gets to, why didn't your flu shot work? Well, how vaccines work is they simulate the exterior of a virus to stimulate your immune system. And so if you can teach your immune system what to look for, then when it finds that real virus, it's going to know how to kill it and what it looks like. And so most vaccines contain markers or the exterior code for many different strains. So scientists try and guess what's going to be the strain this year, make a cocktail of a bunch of them, and do their best to give you immunity against all those different options. But it is just a guess. They've got to run the numbers, see what they can figure out. And because it mutates so rapidly, sometimes new strains that scientists didn't know about will come out, and so you're not protected against it. Or the strains that you are protected against have changed so much that your immune system no longer recognizes it. And so there's several different types of vaccines, but the main two are going to be inactivated or attenuated. So inactivated vaccines are going to be ones that are grown and then killed. And so since it's just protein, we're going to say denatured. So like they'll heat them up or denature them in another way. So they have all those surface markers for the immune system to recognize. But because it's not a whole virus, it's not going to actually be able to cause the disease. The other format is called attenuated. And these are live viruses, but they're grown in a culture. So when they're inserted into a human, they are now very, very weak. So an example is you grow them in very cold cultures so that the virus gets used, their proteins are now adapted to that colder environment. And then when you put them in a human, which is a very hot environment, a lot of the proteins start to denature, they're very slow, and so it doesn't cause the disease, or it would cause the disease, but your immune system works faster than the virus can because it's been attenuated. So two different approaches to the same goal, which is immunity. And then one of the big viruses of the last 30 years is HIV. So human immunodeficiency virus. And this is what's called a retrovirus, which is another really famous type of eukaryotic virus. This, like influenza, has an RNA genome, and it has little proteins called reverse transcriptase. And what reverse transcriptase does is it reads the RNA and then makes a copy of DNA based on the RNA template. That viral DNA is then spliced into that host DNA, and then just like that lysogenic cycle, that's now in the genome, and so that DNA is going to get read and used to make viral proteins. It's going to be used to make viral RNA, and then that's going to get packaged up by the cells and pushed out of those cells in those lipid envelopes, and it's able to infect new cells. Typically, a cell goes from DNA to RNA to protein. So to go backwards from RNA to DNA is kind of heretical to the whole central dogma of molecular biology, and it's really perverting the natural way that these cells work. So it's a really insidious virus. And so HIV is the virus that causes the disease AIDS. And how this happens is HIV will target immune cells as their host cells, so like white blood cells. And as immune cells are taken over, they're no longer able to perform their jobs. As they're killed by the virus, the body is now weaker from fighting off other infections. And so really, it's not the virus that kills AIDS patients, but getting another disease. So when your immune system becomes so compromised, a very common cold can become a life-threatening emergency because your immune system, which normally protects you from the onslaught of bacteria and viruses filling our world, is now so weak that you can't fight it off, and it becomes very dangerous. And luckily, over the years, we've devoted a lot of research to it, and now HIV prevention and HIV treatments, it's not the death sentence it was in the 80s and 90s because we've devoted so much research to it. And this really was, at least in the United States and parts of Europe and a lot of the world, this was a horrible epidemic that we've overcome. But that isn't the same story worldwide. And you can see that there's a lasting legacy surrounding HIV of colonialism and prejudice. Because if you look in places that are not the traditional Western powers or the more developed countries in Asia, you can see higher rates of HIV prevalence. So especially in sub-Saharan Africa, HIV is still a huge epidemic and a huge problem. And we can see that even though the treatments exist, that HIV shouldn't be a death sentence, that or AIDS shouldn't be a death sentence, and HIV should be easier to prevent transmission, a lack in funding and resources as a result of these nations being left in the wake of colonialism have allowed for these rates to still be high. But we can turn this around. We have the tools to do it. We just have to have the political will to help others. And don't think that plants are immune from viruses. Everything can get viruses. So we started with talking about tobacco plants with tobacco mosaic virus, and absolutely plants get viruses just like we do, just like bacteria do. Everything gets viruses. And so typically it'll be really splotchy pigment patterns that are a giveaway. So if you are growing some fruits or vegetables and you notice really bizarre splotchy patterns, that might be a plant virus. And I know a virus, just DNA wrapped in a protein, may sound really simple, but there's actually simpler things that can get you sick that are very reminiscent of viruses. So there's viroids, which are RNA molecules in plants that cause disease, but they don't code for any proteins. So there's no protein there. It's just menacing RNA. And if we go even simpler, there's prions that are protein molecules that have no genetic material, but are capable of causing disease. So we really start to blur the lines between life and non-living, and chemistry and biology, the deeper we explore viruses. So some take-home points for you. Be sure you're familiar with the nature of proteins, that at its core, it's genetic material, DNA or RNA, wrapped in a protein. You should be familiar with the lytic and the lysogenic cycles. You should be able to explain both of those and how they work. You should know the difference between a eukaryotic virus and a prokaryotic virus, that eukaryotic viruses are typically more complex and typically are wrapped in the membrane of the cells they're infecting. It helps them hide from immune systems or other cell defenses. And then you should have a general understanding of how vaccines work, that they use real viruses, either denatured or altered, to stimulate the immune system. So the immune system will then remember what that virus looks like, and when that virus does come along, the response to killing it will be quicker. So those are your big take-home points from viruses. Hope you had a good time and hope you learned something.